Welcome to Med in 5. The goal of our channel is to simplify medical concepts and help you understand them like a jet stream. Today, we break down the mosquito-borne bunyaviruses, which can mess with your brain. Let's explore these questions before we start. Which mosquito-borne bunyavirus is primarily seen in the Midwest or Appalachia? Which one is diagnosed using a zinc smear? Which virus has been eradicated through vaccination? Here we go. Both Crosse virus and California encephalitis virus are enveloped, negative-sense RNA viruses with 3 genome segments and a helical nucleocapsid. The envelope helps the virus enter host cells, but is fragile, easily destroyed by heat, drying, detergents, or UV light. Their RNA is negative-sense, which means the virus must first make a copy before it can produce proteins. The segmented genome can mix with other viruses to create new strains. The helical nucleocapsid is like a protective coat that keeps the RNA safe while the virus infects cells. These viruses can enter the brain, a process called neuroinvasion, and trigger an immune response that can cause brain inflammation, termed encephalitis. Both are spread by mosquitoes. Crosse virus is carried mainly by Aedes tricereatus in the Midwest and Appalachia, while California encephalitis virus is carried by Aedes dorsalis in the western USA. Children are most at risk because they spend time outdoors in wooded or rural areas, increasing exposure to infected mosquitoes. Both viruses take about 1 to 2 weeks from mosquito bite to symptom onset. However, Crosse virus tends to have a slightly longer incubation period on average. After incubation, infection begins with fever and headache, marking the prodromal phase. This stage progresses to a neurologic phase, which in Crosse virus commonly leads to encephalitis and seizures, making it generally more severe, while California encephalitis virus may cause encephalitis only in some cases. During this phase, children may experience confusion, irritability, or lethargy. Severe infections can result in lasting neurologic deficits such as memory or motor problems. Both viruses share these complications. There's no known association with cancer, so long-term malignancy is not a concern. Symptoms of these viral infections aren't specific and may be caused by other microbes, so doctors can't rely on clinical signs alone. Confirmation requires lab tests, usually serology, to detect antibodies. To identify viral RNA in the patient's blood or cerebrospinal fluid, clinicians may use reverse transcription polymerase chain reaction. There's no specific antiviral treatment for Crosse or California encephalitis virus. Management is supportive, including fever suppression, hydration, and monitoring for neurologic complications such as seizures. Prevention focuses on reducing mosquito exposure through practicing environmental control, using insect repellents, and wearing protective clothing to lower the risk of infection in children living in endemic areas. Let's go back to these questions. Which mosquito-borne bunyavirus is primarily seen in the Midwest or Appalachia? Answer: Crosse virus. Which one is diagnosed using a zinc smear? Answer: none. The zinc smear detects herpes viruses. Which virus has been eradicated through vaccination? None. There's no licensed vaccine for these viruses. Got all 3? Beast mode. If this vid helped you understand mosquito-borne bunyaviruses better, help us grow by pressing like, share, and subscribe. Check out the summary table in the description. Thanks for watching. Keep watching Med in 5, your hub for high-yield mini-med lessons.